to the talk.
Thank you. Thank you very much, Jason, for inviting me. And it's my great honor to give a talk here at UCLA as a UCLA faculty member. So today my talk will focus on one aspect of my lab's research, the simulator. And I want to talk about my perspectives on why we need simulators and what simulators can do. So my lab is named as the junction of statistics and biology, which means that we try to bridge the two fields to develop useful tools for us to understand better biology from data. So let's see. I open the laser pointer. Okay. Why do I need simulators? And what are the requirements for good simulators? I think for this audience, computational people, we are very familiar with simulation because when we want to benchmark our new method against existing methods, we need a simulation study to provide some ground truth. But one thing I want to emphasize is that, for biologists, I often heard this criticism: simulation doesn't mean biology. Right? You have worked on simulators for so long. Why don't you study biology? And my answer to this question is a good simulator should generate data that mimic real data. And what does that mean? It means that if the generation or the synthetic data we generate from simulator mimic real data, it implies that the simulator model is useful. And there's a famous saying from George Box: all models are wrong, but some are useful. Indeed, no simulator can really capture the biology, but we hope that if the model is useful or reasonable, it will give us some useful information from the data. So the second goal for using simulator is something that's not often talked about, but I want to emphasize: the interpretation of real data. So if the model is useful, then the model parameter will tell you something. And we will prefer to have the model whose parameters are interpretable, and we hope to use the parameter values we estimate from real data to tell us information. And the third goal I want to emphasize is that if the model parameters are modifiable to reflect some hypothesis, then we can generate alternatives or variants of real data. For example, if you think about the power analysis, we are actually doing this. We try to use the model to estimate what will be the necessary sample size to achieve a certain statistical power. So I think a good simulator should satisfy all three goals. So the data should be realistic. The simulated data should be realistic, so we can say the model is useful. Then given the model, we hope to interpret real data. And finally, if we can modify the model parameters, we can generate some alternatives to real data. So this is a summary of the simulator development in my lab. So starting from 2019, we first published this simulator called RSG Design. So as the name stands, we aim to tackle the experimental design problem. And at that time, we only aim for goal 3. Alternative generation. So it is like a power analysis tool for single cell data. We want to estimate what will be the cell number and what will be the sequencing depth, so we can achieve a certain power for identifying cell types. Then we realized that the synthetic data from RSG Design was not that realistic. And one key aspect was that the genes were assumed to be independent in RSG Design. So we fixed that, and we obtained RSG Design 2, the second version, whose main advance is to capture gene correlations. So this more realistic nature of RSG Design 2 model allowed us to achieve goal 1 better, right? Benchmark. If the data is more realistic, then we will trust the benchmarking results more. And also the model will give us more accurate interpretation of real data because it's more useful. And finally, we move on to RSG Design 3 recently, and it is trying to more data types. So we move beyond single-cell RNA-seq data of discrete cell types, and we can now generate cells in continuous trajectories, and we can generate more modalities like ATAC-seq and protein, and also we can generate spatial transcriptomics data. So more data types for the current version. But all the 3 versions are count simulators, which means that we directly generate the count data, right? 1 count per gene protein per cell. So it's the process data from the single cell sequencing technologies, but they're not the raw sequencing read. So finally, we have this recent read simulator, which allowed us to go from counts to read, so we can do more with the low-level bioinformatics tools. And this method is called SC ReadSim. So that is just our tour for simulator development. And today I'll talk about the two recent simulators, the count simulator RSGDREAM and the read simulator SC ReadSim. So for the count simulator, this is the paper, and this is the goals we can achieve. So this simulator can achieve the 3 goals I mentioned: benchmarking, real data interpretation, and alternative generation. So from these 2 diagrams, you can see that on the left, we try to say that RSGDREAM can mimic real data for multi-omics, right? RNA-seq, ATAC-seq, site-seq methylation, and cells in different states. In discrete cell types, in continuous trajectories, or we can even have the cell spatial locations. And we can also allow experimental designs, including some biological covariates, which might be useful, and the effects we're interested in, or the batch effects, which are undesirable. On the right-hand side, I want to focus on the model. If we trust the model to be useful, given the realistic nature on the left, we hope to use the model to interpret real data, so we can find which genes are differentially expressed between cell types along the cell trajectory across the spatial space. And we can also we hope to use the model to tell you which set of clusters from which algorithm fits the data better, which trajectories fit the data better. So it's like a model selection way using the model. And finally, we can do the model alteration for this alternative generation. That is, we can make some genes to be not differentially expressed between cell types. We can even change the number of cell types. Like the real data may have two types, and we can generate synthetic cells in 1 type. So I would first talk about this RSGDREAM model to explain why we can do these functions. So in summary, the model tries to find an m-dimensional distribution of cells. So the m is the number of features or number of variables. So in the simplest single-cell RNA-seq data, m is the number of genes. And we want to capture a distribution of cells. So when we generate synthetic cells, we just need to do sampling from this distribution. So these notations are for the general purpose. So they can refer to single-cell RNA-seq data, ATAC-seq data, or methylation data. So specifically, we consider the real data to have n cells, and we use I to denote cell I, J to denote feature J, like gene J. So Yij is the count we measure in the real data. And this X matrix is what we call the cell state covariate matrix. So in other words, for every cell I, we have a covariate vector. So if it's just a cell type, then this covariate is just one-dimensional, categorical. And if it's pseudo-time in 1 trajectory, it is one-dimensional, numerical. If it's multiple trajectories, say p trajectories, then this covariate vector is a p-dimensional vector. Or if it's a spatial coordinates, right, 2 dimensions or 3 dimensions, depending on the data, but typically it's 2 dimensions. So then this state vector will be 2-dimensional spatial coordinates. So we want to model, in our model, we'll consider the state covariates like the cell type, cell pseudo-time in trajectory, and cell spatial location, how these covariates would affect the count we have in Y. And finally, we have the cell by design covariate matrix. So here, every cell have a design covariate vector. So specifically, we consider 2 covariates. One is the batch, one is the condition. So each is a categorical variable. So we want to allow for batch effects and condition effects to make to model more complicated designs in real data. So first step of our modeling is try to model the marginal distribution of every feature given the state covariates and the design covariates. So specifically, we talk about feature J. And we use this very general model called GAM-LSS, so standing for Generalized Additive Model for location, scale, and shape. So this can be considered as a generalization of the generalized linear model. And the generalization happens in these aspects. So first of all, this Fj stands for the distribution of feature J. It is depending on Xi, the state covariates of cell I, and the design Zi, the design covariates of cell I. So given them, the distribution will be parameterized by the mean, by the standard deviation, and by the zero inflation proportion. So for each of the three parameters, we don't have to have all of them for other models. I will show you next slide that Fj has many choices. But this is the most general case that we will have three parameters. So for each parameter, we will have a link function, just like in the generalized linear model, so that after this link function transformation, on the right-hand side, we can model the effects of the state covariates Xi and the design covariates Zi. Just for information, Zi is already decomposed as Bi and Ci for batch and for the condition. So you can see in this link function, each link function on the right-hand side, we would have the batch enter as an intercept, condition enter as an intercept, and then given the condition, the effect of the cell state Xi can be nonlinear. So linear is the simplified form, but it can be allowed to be nonlinear. So I will show you the forms, the nonlinear forms we'll consider for each type of data in the next slide. So if you want to understand more about GAM-LS, actually I have taught a class here, Stats 205, for our graduate students for about generalized or hierarchical linear models, which covers generalized linear model, which is the foundation of GAM-LS. If you're interested, because of the pandemic, so my lectures are online. And so for the model, Fj, the marginal distribution for each feature J, given the state covariates and the design covariates, we allow multiple choices, including Gaussian, Bernoulli, Poisson, negative binomial, zero inflated Poisson, and zero inflated negative binomial for different modalities of data. For example, for single-cell RNA-seq from, say, 10x genomics, then negative binomial is known to be a good distribution. And if it is like single-cell methylation data, you may want to use Bernoulli distribution for the binary data. And for some other modalities, you may want to use Gaussian. So you want to be flexible for the choice of distributions. And you can see that for each distribution, we have different numbers of parameters. But for the most complicated, say zero inflated negative binomial, we have all three parameters: mean, standard deviation, zero proportion. And for that nonlinear effect of cell states, we consider these options depending on the covariate type of our states, right? It could be a cell type, then the cell type effect will be just additive, just an intercept. If it's like one lineage, then we use this spline function to model the nonlinear relationship. If it's P lineages, we will have a spline for each lineage and then add up. And if the state is the spatial location, we actually use the Gaussian process to model the effect of spatial location on the gene expression mean. So that's the marginal part. We will do the modeling for each feature separately. And the key part is to do the joint modeling. And for this, we actually use the technique called Gaussian copula, which try to couple the M marginal distributions, each is one-dimensional, into an m-dimensional joint distribution. So this is a simple cartoon to describe what we do exactly. For the top, and we just visualize two features as two genes for simplicity. On the top, it is the marginal modeling, which I already described using the GAM-LSS. But this is a simplified case for just using negative binomial. The bottom is the joint modeling part. So given the marginal distribution, we will convert each cell's value in that dimension, in that feature, using the feature's marginal distribution into the CDF value. So cumulative distribution function value, which we can obtain given the value and the distribution. So with the CDF values for all the features, what we did is that we convert each dimension further from the CDF values to standard Gaussian values. So to do this, we will use the inverse of the standard Gaussian CDF function to achieve that. So like an inverse distribution transform. With those standard Gaussian values in each dimension, what Gaussian copula does is to fit a multivariate Gaussian distribution because every dimension is now Gaussian. Then this is our joint modeling. From here, so this is the breakpoint about the fitting to real data. So to the right, we will do sampling, that is to generate synthetic cells. So we first sample from this multivariate Gaussian distribution. Every sample point will be a synthetic cell. And every dimension is still in the scale, on the scale of standard Gaussian distribution. Then what we do is that to use the standard Gaussian CDF to convert each dimension into CDF values. So this is what we have here. And with those CDF values in each dimension, now we will use the marginal distribution we got on the top to do the inverse distributional transform. Then each CDF value will be converted to the corresponding quantile in this marginal distribution. And that will give us the values in the original scale as the real data. So in other words, we will use this part, marginal and Gaussian copula, to approximate an m-dimensional joint distribution, and every synthetic cell is sampled from this fitted joint distribution. So if you want to learn more for our students about GAM and copula, there are two very good books to introduce those two classical techniques if you are interested. And the R package for GAM is also very well written. So I will continue to talk about what we can do, right, with the RSGDREAM. So about the 3 goals I mentioned in the beginning. First, we want to do benchmarking. That will require us to generate realistic synthetic data. So this is a simple comparison with another simulator, which is deep learning based, RSGAM. So we want to argue for 2 things. First, we are very fast, much faster than GAM, and then this package, I should say. And second, this value, which is from 1 to 2, it measures the similarity between synthetic cells and real cells. So 2 means perfectly similar, 1 means dissimilar. And we have higher values, which means that our synthetic cells are more realistic. And this is to show what we can do for spatial data, 10x Visium. So top is real data, bottom is our synthetic data. And this is for site-seq. So for every protein and its coupled RNA expression, CD3, CD8, CD14, we want to show that the top is the real data, bottom synthetic data. We can mimic the real data pattern very well, including the relationship between protein and RNA. And with the realistic data, we can do benchmarking. So this is the result from our paper showing that we can use RSGDREAM to benchmark the tools for spatial transcriptomic cell type deconvolution. So this is not a comprehensive benchmark, but just a demonstration of what we can do. So we consider 3 tools, CARD, RCTD, and Spotlight. So based on the result, we show that CARD and RCTD perform similarly in our benchmark, and they're better than Spotlight. So that's goal 1. Goal 2, if you trust our model, what can we do? And we want to say that with the model fitted to real data, the model parameters can give us useful information. So the Gaussian copula, what it can provide is a pairwise gene-gene correlation matrix. And this correlation matrix, even though it's not on the original scale, but in the transformed Gaussian scale, but it still offers very useful information. So one thing we can see is that if we fit a separate model on each cell type, so it's a cell type specific model, we can see the gene-gene correlation matrix are more similar between the 2 T cell types, regulatory T cells and naive cytotoxic T cells. So their correlation matrices are more similar, but they're very different from the monocytes and the B cells. And also, we can provide a way to summarize this correlation matrix into a vine structure. So I won't go into detail of the vine, but basically it is like a hierarchical simplification summary of the correlation matrix. So from this vine, like a tree, we can see which genes are core genes that are more correlated with other genes. So this will also give us useful information, like in a network analysis. So for goal 3, alternative generation. So I want to say that one use we have implemented already is to generate one hypothetical cell type as some null data given the real data, which may belong to two cell types. So what we try to preserve in the RSGDREAM model is per gene mean, per gene variance, and gene-gene correlations. So one application of this synthetic null data is to try to address this double dipping issue in post-clustering DE analysis. So this is known as double dipping because we use the same data for twice, first to cluster cells and then to test which genes are differentially expressed DE between cell clusters. So this is a toy example to show that if the cluster results are spurious, that means you just arbitrarily set a boundary. Then you will find the genes differentially expressed because the boundary is set because of the genes. So this is like a confirmation bias. You use the same genes to define clusters, then of course some genes will be naturally DE. But that doesn't mean your clusters are trustworthy cell types. So to avoid this issue, we set up the synthetic null data in the beginning of the analysis. So before you do clustering, before you do DE. And our goal is to remove the false positive cell type marker genes because our synthetic null data only contains one cell type. So that means no marker genes should have been found from synthetic null data. If there are, then these are the genes you're not so interested in. So for every gene, we do the same analysis clustering DE, and the gene will receive a score about how likely the gene is a DE gene. So the score from the real target data and the score from the synthetic null data, they will be comparable. A gene is only a trustworthy cell type gene if the target real data score is much higher than the null score. So we can do a comparison at the end of pipeline. And we have a technique called Clipper, which was already published for controlling the false discovery rate in this contrastive strategy. And this work was led by my former student, Shinzhou, who's now in Oregon State. So just to quickly show the results. So the first interesting finding is that using the synthetic null idea to remove the confirmation bias in our analysis pipeline due to double dipping, we can better distinguish cell type markers from housekeeping genes. So this is one typical marker, CD16, because we're looking at CD14 monocytes and CD16 monocytes. So this is a well-known marker, and this is a well-known housekeeping gene. So in our results, the two genes ranks differ a lot. Housekeeping gene ranks low, this marker gene ranks high. But in Surat, they're not so distinct. The reason is that the housekeeping gene has a high variance that can likely drive the clustering. And this one shows that if you look at the top DE genes we found by cluster DE and top DE genes found by Surat, then our top DE genes are more distinct between the two clusters. This is also due to the reason that we removed the genes using the synthetic null data. And this is the gene set enrichment analysis, where we try to compare two sets of genes, known markers and known housekeeping genes. We want to see where each set is enriched in our ranked DE gene list. And we can see that the marker genes are enriched in the top, while the housekeeping genes are enriched in the middle to bottom. While in Surat, their distinction is not so obvious. So finally, I want to say why we have this result. The reason is we do the contrast. We have the target DE score, which is the only decision criterion Surat uses, right? It tries to use target data only, real data only. But once we contrast, we subtract the null DE scores, then our contrast scores, the y-axis, can better distinguish these two sets of genes. But if you look at the target DE scores only, the distinction is not as clear. And this is another real data application to show what cluster DE can help with the real data analysis. First, from this Drosophila Visual System Developmental Atlas dataset, we actually spotted 3 pairs of clusters, and in each pair, the 2 clusters are not so distinct. So that's why in the original publication, even though there are different clusters, but the authors didn't give them distinct labels. And using our cluster DE, we see that we found 0 DE genes between each pair. So this will suggest that we would encourage those clusters to be merged. And this plot further confirms that, yeah, for each pair, the two clusters are really not distinct in the UMAP space. And furthermore, we want to say that using cluster DE to find the more reliable cell type markers, these cell type markers can better distinguish cell types that are truly different. So between these two cell types, these are the top DE genes found by cluster DE. So you can see they are indeed more distinct between the two cell types than the top DE genes found by CIRAD. And this is confirmed by the classification accuracy. So using our top DE genes as features versus using the CIRAD top DE genes as features. So this is the preprint of our paper, Cluster DE. So we welcome your comments and your suggestions because we would like to have this method be useful to add some synthetic null data to your real data analysis. So that is the count simulator. So I will talk about our read simulator in the last part, right? The final stage, read simulator. So this one was recently published, SC Read Sim, and the work was led by my student Guang Ao. So this is what we try to achieve with the read simulator. So here we are not so capable of doing the real data interpretation for the read generation process because that process is at least for now we think it's like a sequencing experiment, and so there are some parameters, but we just try to mimic real data without trying to summarize further. So I would say the read simulator can achieve two goals so far. One is benchmarking because we aim to generate realistic data with some ground truth. And the other goal is alternative generation. We can allow you to modify your model so you can generate alternatives to real data. So the goal of the read simulator is to allow us to benchmark the analysis tools that work with reads, not just counts. So previously we look at the counts only. So I want to quickly show that what SC Read Sim can mimic, what characteristics that it can mimic in the real data. First, K-mer frequency and sequencing error rate. So these are all features, characteristics at the read sequence level, and we try to mimic real data well. And second, oops, I think it's stuck. Sorry. Okay, and this shows that we can also mimic the read coverage in real data at the cell type level. So we can mimic the read coverage for specific cell types. And furthermore, using the realistic data, we can do benchmarking. So this is just one set of benchmarking results for peak calling on single cell ataxic data. So our result shows that using our ground truth peaks in the simulator versus the non-peaks, so the read coverage is better preserved by two tools, MAX3 and HMM attack. So they have better resemblance to the ground truth peaks. So their COD peaks have better resemblance to the ground truth peaks than Seeker and Homer. And so this shows the ROC curve and precision recall curve. So we find the top performance in the benchmark. And for alternative generation, the third goal, we can allow users to design ground truth peaks. So for example, this is the ground truth peak, and this is something like a peak in real data, but we are not so sure. So we will try to wipe out such information for removing ambiguity. And also here, right, there is a small peak here, but if we don't want to say it's a ground truth peak, we can remove it, or we can keep the strong signal to give us some cleaner ground truth. And I think the computer's running slow here. We can vary the sequencing depth. So you can generate data with more reads versus less reads. And this can help us evaluate how robust a method performs given the sequencing depth. And also similarly, we can change the cell number. So to summarize, I talk about our recent developments on simulators. And this is a summary of the three goals I think a good simulator should aim to achieve. Benchmarking, which requires realistic simulated data, and that will tell us backwards that the model is useful. So then given the useful model, we hope to use it to interpret real data. And finally, we want to generate alternatives to real data for achieving more, for giving us more information about like the bias and the stability of our analysis results. So our ongoing work has two main projects, just to quickly give an overview. One is to continue this route to help us generate some synthetic data and synthetic null data for spatial transcriptomics. So this is one analysis for spatial domain detection. So we want to find some negative control, that is, the spatial domains are not so distinct as a control to the real data where the boundaries are very distinct. So we hope to provide something similar to cluster DE for spatial data analysis. And another direction is to generate population level single cell data. So this is the preliminary result to show that if we generate single cell data for many individuals from their genotype data, then we can do better than the current only existing simulator SplatPop. So you can see that the UMAP plots, we are doing better, and the MILSI values are higher for us. So we also hope that to show, okay, our model can generate more realistic population level data. That implies the model can give us better inference estimation of the SNP gene expression relationship at the cell type level. So finally, I want to acknowledge my members. My student Tianyi, who is actually defending today, he developed the second version, which I consider to be a big advance from the first version, because we can go from just Go 3 to both Go 1 and Go 2. And Dongyuan, who go further to develop this third version, current version I talk about, and Guangao for the race simulator. And these people, Dongyuan Kexing graduated who left for Microsoft, Christy who's in the audience, and Xinzhou who left for faculty position Oregon State for their contributions to cluster DE. And my visiting student Siqi for leading the efforts, ongoing efforts for spatial transcriptomics, and Chris for the population level data. And my funding agency support for their support. And also my student Dongyuan is on the job market this year. So I'm very glad to have him as my student. Thank you very much.
We have time for questions.
Hi. Yeah, it's a great tool. I read your paper carefully. So just to you have some questions about your spatial simulation part. So, so far, like in your presentation, all your examples are from like a Visium or old ST. So, so have you tried like simulating like a single cell resolution, like a Merfish data?
Actually, in our paper, we have that. In our paper, we do have that. Yeah. So, but I think for now, that simulation is not as ideal, to be honest, as for the 10x Visium data, because in our simulator, if you look at the link function I talk about, we use Gaussian process to model the effect of spatial coordinates to gene expression. So that implies some smoothness. But I think for Merfish data, it's more like sporadic. Right, so you will have the spots, which are the detected genes, instead of capturing like a smooth average across cells. So I think there's definitely room for improvement for single molecule spatial data.
So I think I wonder why you chose like a Gaussian process model for. I think could be because okay, I'm giving the next presentation and I will show example of negative spatial autocorrelation in like Merfish data. So in that case, Gaussian process might not work very well. But I wonder what if you use like a CAR or SAR model for the spatial simulation.
So that's exactly what I explained. That is, we use Gaussian process for Visium data, right? So because Visium is observed to be smooth, and I acknowledge that we are not doing so well for Merfish data. So in our paper for Merfish data, we have to make the Gaussian process complexity to be very high so that can capture those sharp changes in the space.
All right.
Yeah. So I think it's a room for improvement. Yeah, for sure. Yeah. Thank you.
Other questions?
I have a small question about how many gene you use for simulation, because one slide that shows you using 1,000 gene.
That's right.
For that. So if we make the number of gene being larger, it will kind of help the mimic the cell, or it will give not good effect. I mean, how many genes is good for the simulation?
I think to clarify your question, we need to define what is good.
Yeah.
Yeah,
sure.
yeah. I think to say, if in the ideal case you want to simulate all the genes in the real
Yes,
data, for
yes.
sure. And I think for that plot, I think the SC Design 3 versus SC GAN, it was just for fast computation. We use like 1400 genes or something. And I think it depends on your analysis goal. But if you look at the data, for sure, many genes have very low expression, right? Close to zero counts. So we are still trying to make our simulator SD93 run faster. But one aspect to speed up the computation is to consider not so many genes in the joint part, in the Gaussian copula part, because that is time-consuming step, because it's the square of the number of genes, right? The pairwise gene correlation. So for the genes that are so lowly expressed, it doesn't make much difference if you include them or left them out from the correlation estimation. Actually, they will hurt the correlation estimation if you have more genes. But you still want to have the marginal distribution for every gene. Yeah, so that is something tunable, right? Number of genes you want to simulate and the number of genes you want to consider for correlations in the joint distribution.
So can we change that in the package, because I'm using that now? So
yeah, we are working on that. So our next step is try to make the package modular. So like a Lego. So you can basically piece modules together for your simulation purpose.
so in that case, we will use the cell type space, not cell type specific, some high express gene in specific cell types, and use them to calculate the copula.
That's right. Yeah. I think we would like to have that function, so you can decide which genes you want to include in the copula and which genes you just want to do the marginal modeling. Yeah, thank you.
So when it comes to like improving the spatial simulation, so you just remind me, for you're already using GAN model, so somewhere relevant is there's the Moran eigenmap, there's spatial filtering, and from Moran eigenmap you can use like the components for a negative spatial autocorrelation, which might help with the Murphish simulation, but meanwhile like that might not be very scalable. So
So sorry, that is a question or a comment? Yeah. Okay, so thank you for the comment. Yeah, but we can talk further. Yeah.
I have a question.
Yeah.
So, I mean, you had mentioned like one of the competing methods was the GAN.
Yeah.
So have you sort of asked from the perspective of like, can you train machine learning methods to discriminate your simulated data from real data? And is it easier to discriminate it versus like the GAN type outputs?
Okay.
And if it is able to discriminate it, do you know like what it's sort of learning to discriminate your simulated from real data? Right.
So in our simulated data, we didn't have that GAN structure for doing this computation because we're just doing a model fitting. And I think for the SE GAN, maybe the model, because it's not our implementation, someone else's implementation, it's very possible that it was not trained to the optimum. So that's why it's not as realistic as we hope it to be. But on the other hand, I think for goal one, I think the GAN idea should be reasonable if you want to aim for realistic nature for the synthetic data. But I think for the goal 2 and goal 3, interpretation and model modification, I don't see how easy we can do with the GAN. Yeah, so that's why we try to advocate for this model. And also I want to say that this model was built upon many empirical evidence, like negative binomial distribution was observed to work well for the UMI counts. So those kind of knowledge help us. And also the current Gaussian copula only considers pairwise correlation of genes. So it's like a second order interaction, but we didn't consider higher order interaction. Whether that is necessary or can be further improvement, I think that's also an open question.
Is there an online question?
An online question.
Sure.
So thank you for the wonderful tools developed at your lab. I've been using cluster D for a while.
Oh, thank you.
And wondered if you have any plan to improve the speed of it for large scale.
I think so. We do have the plan. Yeah. I would say we do have the plan. Especially, I think the current time limiting step. But maybe the person can try the newer version because we just updated the package. Yeah, we just have the update. I think the major improvement is about the synthetic null generation part. Yeah. And I have to admit that even though we aim to have RSG Design 3 to be more general, but in its first implementation, for the same tasks that RSG Design 2 aim to do, like the discrete cell type, which is a special case of RSG Design 3, RSG Design 2 is faster. So that's why we still have like software implementation to improve. But I think we did have some improvement in the current version. Yeah. Because yeah.
Okay.
Yep.
Okay, so
Do we have time? Sorry.
a quick question.
Oh, actually, it's not a big one. I was thinking, how do you think about mechanism, right? And I
Yeah.
think the design is getting to that question.
Yeah.
Like incorporating the design aspects, but then they're getting deeper into mechanism, like, embedded dynamics.
Yeah.
How do you see?
Yeah. I know there is a simulator, to my knowledge, from several labs that use a different perspective, right? They use like differential equation based on gene regulatory network to simulate single cell. And I think the most recent work is from Shiwei Zhang's lab at Georgia Tech, and she has a bioarchive preprint. And I think a very interesting open question for the field is that how can we bridge these two perspectives? One is like first principle driven, mechanism driven simulation. One is like data driven modeling. So I think for us, we focus more on mimicking real data, but for them, they focus more on starting from some ground truth that's in the mechanism. So can we bridge the two in some way so we have a in the middle, right? So we can have the data more realistic while also start from some principles. I think that is a grand challenge, because if you can really do well, like if you simulate from the mechanisms and your data are very realistic, then you will say, oh, the mechanism you have, your hypothesis is more likely true.
So those simulators are not generating good quality data.
Or I would say mimicking real data is not their strength.